Welcome to this video podcast asking the question, does exercise negatively impact reproduction? And to start this off, this is in horses. So this is not meant to be medical advice for women, but what we know in exercise and effects on reproduction, much of that information has actually been done in humans. So has been looked at in males and females. Now we are looking at some of that impacts on other athletes like the horse. And that is the whole purpose of this discussion, is to look at the impacts that exercise might have on reproduction in the mare particularly, and then maybe the stallion, and we'll save that one for the end. So what is the genesis of this? Asking the question, can exercise impact reproduction negatively? And as a young PhD student, when you start off in your career, you're hoping to contribute to the field of science, but don't really expect your research to have much of an impact, especially early on in your career, because you look up to those old professors, those mentors that I've had through the years, Dr. Ann Rodick, Dr. Martha Vogelsang, many others that have impacted me in my career. And you hope one day to equal them. And so this genesis of this research actually started with Dr. Vogelsang when I showed up to Texas A&M as a bright new PhD student, ready to go, aggressive, want to learn as much as possible. I had a very keen interest in embryo technologies, these advancements that we've been making in cloning and embryo transfer, oocyte transfer, all of these techniques that we now use in the modern day breeding barn with our horses and many different breeds and disciplines. And Dr. Vogelsang, when I showed up, told me she had the perfect project for me. And that was heat stress in mares and impacts on reproduction. Her husband, Stephen Vogelsang, was a broodmare manager or a farm manager at a very large embryo transfer facility in Central Texas. And embryo transfer, and this is early 2000s, so 20 years ago, but embryo transfer in the late 90s, going into the 2000s, had become more of a technique horse owners were using for their mares. And the reason of that is because the AQHA, who is the large breed organization in the United States, American Quarter Horse Association. And there's over 4 million quarter horses around the world, millions in the United States. And the AQHA breeding rules used to be that a stallion could breed as many mares as he could throughout the year using artificial insemination. So theoretically, a stallion produces billions of gametes. So you could produce as many foals as you could on that one stallion. And then you could come back and register that foal to that stallion. So typically some of these stallions had hundreds of foals, the very, very top ones registered to them, where mares could only have one per year. Now, genetics, 50% of the foal is the stallion, 50% is the mare. The mare is just as important in the genetics of that foal. And so it was a bit unfair, unfair to the mares. They could produce some top foals for good money for a lot of these horse owners, but yet they were restricted. Now, there was a lawsuit, don't need to get into all that, where the AQHA did open up their rules to where the mares could breed and produce as many foals as they could per year using embryo transfer. Now, to back the bus up a little bit, embryo transfer is you generally artificially inseminate the mare, and then she ovulates, and then fertilization takes place within her. And then at day 7, you can go in and flush her uterus and get the early embryo, which is just a big clump of cells. And you have to look with a stereo microscope to find them. But then you can take that early embryo, it's spherical, and then transfer that into a recipient mare. And the recipient mare can carry that pregnancy to term, nurse the foal, raise the foal, and then wean the foal. But that foal's genetics goes back to that stallion and original mare who donated her egg, right, and fertilized the egg. So that has allowed embryo transfer to grow rapidly in the last few decades in the horse industry. And I just use the AQHA as an example. Now, Jockey Club are racing thoroughbreds. It still is El Natural. There is no embryo transfer going on there. It's not allowed by their breeding rules. But with our mares, we can push them a little bit reproductively. And on average, mares can maybe produce 4 foals a year. You don't want to push them too much. They still have to cycle, and you still need to go in and flush their uterus. So you don't want to do that too much within a year. And I know there was a record. It was at a meeting I was at in Argentina where they got over a dozen embryos from a mare in 1 year. So that was really pushing her hard. Typically, we do about 4, and then we'll let her rest until the next breeding season. But that has allowed embryo transfer to grow. Okay. So going back to heat stress and Stephen Vogelsang, he had noticed that mares in late spring, early summer in central Texas, where it gets really hot during the day, were producing less embryos, and they were also of poorer quality. Now, to caveat that a little bit, and we didn't know this at the time, but he thought it was more of a heat stress issue, because we do know heat stress, which we covered in a previous podcast and video podcast, can negatively affect reproduction in other species, like cattle's the big one. So Dr. Pete Hansen, he was one of my colleagues at the University of Florida. He is well known around the world for investigating heat stress in dairy cattle and their impacts on reproduction, negative impacts on reproduction. So I actually, before I even went to Florida, I was involved, reading a lot of his research when I was setting up my PhD dissertation work. So with embryo transfer, the other benefit for these mares is they can continue their competition schedule, still train, still be ridden, and go out and compete. So you have some brood mares that are just brood mares. They're done competing. They're just producing foals, and they're involved in embryo transfer. Then you have these competition mares that in their competition careers can produce embryos and produce foals through embryo transfer. So we didn't know at the time. We didn't know if heat stress impacted horse reproduction, and we didn't know if exercise could impact reproduction. But Stephen Vogelsang said there's something there. And then Dr. Katrin Hinrichs, who I worked with at Texas A&M, world renowned. She was the one really heavily involved in horse cloning. Incredible mentor too to me. She also said, yeah, there's something here. And she came onto my committee and helped me through this project with Dr. Martha Vogelsang. So we started to look into heat stress in horses. Now, horses, as we talked about in the previous podcast, they can suffer heat stress in really extreme conditions, but generally, as long as they have shade and water, they thermoregulate pretty well. During a hot summer day, when you drive around and you live near livestock, you can see cattle ruminating under the shade, typically, and horses out there grazing in the sunshine. Because they can go, if they get hot, they'll go to the shade or they'll go drink some water and cool off. They're sweating a little bit, but generally they don't get too hot. And when we looked at setting up this experiment, or the series of experiments that we did, we thought, okay, if we really want to heat stress them only, we're going to need environmental chambers. And there's a lot of concerns with putting horses in environmental chambers. We didn't have them at Texas A&M, and welfare concerns of putting horses in something like that. It is done. There is like oxygen chambers that people put horses in for healing and other things. So it's not something new to the horse industry, but when we looked at doing environmental chambers, we decided no, that's not our best option. And again, we're doing scientific research under veterinary care with permission from the animal use committee at Texas A&M. That's a completely independent organization that evaluates any animal research, and if there's any stress or welfare concerns, you have to address it. And then if there is, as part of the experiment, you have to really argue why you need to do this experiment. And a lot of times you can't. So at the university level in the United States, and I know in many parts around the world, animal welfare is a top concern. Okay, so I just wanted to put that there. So the next question was, okay, how are we going to get these mares hot? And in the other podcast with heat stress, I did cite the gear study. That was like the basis. I know I gushed over it because I do remember reading that study and going, aha, here we go. This is how we can elevate body temperature. And that study was heat storage in horses during sub-maximal exercise before and after humid heat acclimation. So when their horses were exercised between 90 to 92 degrees Fahrenheit, 85% humidity, after 20 minutes, their core body temperatures spiked to 106.7, which is about 41.5 degrees Celsius. That is super hot. And few of my horses got in the 106 range when I did my study, which I'll talk about, but not that hot. We didn't typically see them get that hot because Texas in the summer, humidity varies throughout the day. Generally, it's higher in the morning because the moisture's in the air. But by mid to late afternoon in Texas, it's mid humidity, about 50%. Still hot, very hot, but not quite as humid as, say, somewhere in Florida, where a rainstorm will come in, dump a bunch of moisture on the ground and in the air, and then storm goes off, and then it gets up to 95 degrees, and it's really humid because the sunshine is evaporating all that moisture, and it's in the air. That's the humidity part. Well, in Central Texas, College Station, it's more dry. So I did approach my committee and said, okay, I think we can spike the temperatures in these mares by exercising them. And we know heat is a byproduct of exercise. So it wasn't really a heat stress study effects on reproduction. It was more exercise stress under hot and humid conditions. So, or hot conditions, maybe not humid all the time. I mean, 50% humidity, 60% humidity, you can argue is still pretty humid for certain parts. But the idea was exercise stress. And when you look at how or hypothesize, okay, I'm going to exercise these horses. I'm going to spike their body temperature. So in my head, I'm thinking, okay, am I going to cook that early embryo? I mean, you're talking just a few cells to a few thousand cells. Now, the horse's body is trillions of cells. Early embryo development, it's a single cell first day, then 2 cells, then 4 cells, then 8, 16, because they're dividing, 32, 64, day 3, day 4. Then you get into the blastocyst and you have thousands of cells. It takes a long time for that horse to get to the trillions of cells. But I thought, okay, if we're heat stressing them, maybe that we're cooking them, or the oocyte from the mare, her egg, gets damaged due to body heat, or the sperm cell from the stallion after insemination and exercising her. Maybe that's contributing to reduced fertility. Because again, this idea that it does reduce fertility, the heat and then the exercise. Now, also what I talked about in the heat stress video podcast, when a horse is exercising, and even us, blood is shunted away from the core, because the core is where the muscles are working, the heart's working. You're getting the muscle byproducts is heat, and blood is shunting to the extremities to cool the horse off. So the body is going through some stress and in that fight or flight mode, because that's what exercise, it's flight, right? It's kicking in the stress system of the horse, and it's shunting blood away from the digestive system, from the reproductive system. The last thing a mare wants is to be reproducing if she's stressed or running for her life from a wolf or something in the wild. So it makes sense that the body's set up that way, that, reproduction is one of those things that start to go with stress, especially chronic stress. Acute stress you can overcome relatively quickly. It's the chronic day after day after day, which a brood mare that is in competition schedule, in a training schedule, she's going through those daily stressors of, being handled, being groomed, being tacked up or whatever, going through stress to train and compete, right? That's like our prime athletes in humans. So when you start to divert blood away from the reproductive tract, you are diverting energy, nutrients, oxygen, hormones, critical. Because whenever I talked about reproduction, because again, my PhD is reproductive biology, I always describe it as a series of carefully orchestrated events to be successful. There has to be step 1, step 2, step 3, step 4, step 5, in all these different orders to be able to get a mare to conceive a foal. So the mare has to develop follicles on her 2 ovaries without getting too much into reproduction. But that takes hormone signaling plus nutrients. And then, as you're going to see in a follow-on study I did, blood flow to those follicles that are developing on her ovaries that house the eggs. And then she's going to ovulate one of those. And that's hormone signaling. Then the oocyte travels down into her oviducts, or the fallopian tubes, which you may be familiar with. And the sperm cell needs to be there to meet it. The ova has about 8 hours. After it's ovulated, you roughly have about an 8-hour window where it needs to be fertilized or it's done. It dies. And then once you get fertilization, you have a 1-cell embryo, very, very sensitive, for about 24 hours. And then again, it goes through that 2, 4, 8, 16, blah, 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 division over the next following days. And there needs to be oxygen in the reproductive tract, nutrients, and it has to be at an appropriate temperature, because if it gets too hot, it can injure those cells or kill those cells, and then the embryo's no good. So that is what we were hypothesizing. And then when you look at the hormone signaling, the brain's releasing stress hormones. Well, typically the brain's releasing what we call FH or LSH, which are signaling hormones for the mare to develop follicles and ovulate. So when the body's stressed, those hormones are impacted. They're not released as much or as in greater quantity. The stress hormones are, and that's what the body's responding to. So you have all of this competing with the body to keep the horse alive, right? That's the whole purpose of the stress response system, is to escape predators or survive whatever stress it is and then heal and overcome. And that's what training does. That's what exercise does. And then with heat too. Heat also can stress the horse, as we previously talked about. So that led to this project that we set up. And my committee was obviously Dr. Martha Vogelsang. I had Dr. Katrin Hinrichs. I had Dr. Dewey Kramer, who's like one of the grandfathers of embryo transfer. He was the big cloner in the early 2000s. Dewey, the deer, was named after him, the first clone deer. Cece, copycat, the cat, that was his, did well, lived a full life, that horse, or that cat did. I had Dr. Nancy Ng as a geneticist. Dr. Young Ho Choi, who was the ICSI guru of the world in the 2000s. That is in vitro fertilization for the horse. Whole nother podcast, different topic. So I had a really robust committee guiding me, and some incredible people that I got to work with, alongside with, and they set me off on my career. That resulted in my first published paper in 2009 titled Embryo Recovery from Exercise Mares. And I love that paper because it was just, I didn't realize that research would have such an impact on the industry. As a PhD student, you just never did. All right, so how we set this up. So we were going to exercise the horses in early afternoon. So we had a mechanical exerciser. So there's a mechanical hot walkers. If you haven't seen them, they have a series of gates and you can set them and it can push the horses to a trot, light canter. I would say our exercise was moderate. It wasn't strenuous. We weren't pushing them too hard. And we did it 30 minutes. And then we had the cool down, which I talked about in the previous podcast. Had a couple horses come up heat stressed. We cooled them down with hoses, walked them, and we would never return them to pasture until they were properly cooled down. So this wasn't extreme. The average temperatures over the study of the 2 years I did it was 86 degrees Fahrenheit. It's about 30 degrees Celsius and about 50% humidity. So this wasn't extreme. And again, we monitored those horses very, very carefully. We checked their temperatures all the time during exercise, after exercise, and then after that cool down period. And again, they would not be returned to pasture until those temperatures were getting back close to baseline. So I think that's what you find interesting. This wasn't extreme. This wasn't heavy or very heavy exercise for the horse. These are brood mares. They were given training before this to be fit. So they were fit, but these aren't or weren't competition horses. Now we artificially inseminated them. And then on day 7, I went in to collect an embryo. And Stephen Vogelsang taught me with Dr. Catherine Henricks on my method. I would go in and you set up a flush media that's commercially available that goes through tubing into the uterus of the mare, which has a balloon at the tip of the catheter, which creates kind of a vacuum in there. And I would put 500 milliliters in there. I would go in and massage that up because the mare has 2 uterine horns, because you don't know where the embryo is at day 7 if it's in there. And then drain it, goes through a tube into a collection dish, which has a 75 micron filter. So the embryo wouldn't go through, but the fluid would. And we would drain that out. And I did that 4 times. Then I would take that into the lab, put it in a collection dish, and look for that embryo, if there was one. Now, typically in embryo transfer, you would take that embryo and go and put it in a recipient mare. Our embryos were used for genetic analysis to look at any damage or heat stress that may have experienced in a later on study. But I did this over 2 years. And again, the results were really striking. We just didn't expect it, didn't anticipate it. And, again, I go back to my mentors at A&M. Just to give you an idea of how hot these mares were getting in exercise. So in that paper I published, the initial temperature was on average 37.96, which is typical for horses. Then at the end of exercise, the average temperature was 39.93, and we had a range of 39 degrees Celsius, which is about 102, up to the maximum temperature I got any of those horses in the study was 41.28 degrees Celsius, which was 106.3. And that was just a few of them. Most of them were in that 103 to 104 range, looking at that. Now, when you do this research, you look at all the data you can. And this is what I found really interesting. I remember the day doing this too. I was in my apartment and all the statistics, and you do all your spreadsheets. And then I started looking at averages, and I'm looking at every little thing I could. And I remember when I found this data, I was like, holy smokes, this is incredible. So our horses that were exercised had a delay to ovulation, and they ovulated smaller follicles. Okay. So when you looked at their ovulatory, from ovulation to ovulation, our horses that were exercised, their follicles did not get as big and it took them longer to develop. And the hypothesis is because of stress, that disruption to the hormones, it took longer for that mare's reproductive system to ovulate. And you can hypothesize a whole bunch of different things. I have a little bit more data that backs up a little bit of this, thinking that oocyte that's ovulate is probably not a great quality oocyte because it has to be properly matured in that follicle. It grows, it gets this cumulus layer, it needs the nutrients, it needs the oxygen before it's ovulated. And exercise appears, based on just this data, and that it's delaying that process. And then the follicles, when they ovulated, were smaller. So that was really striking data. And human research has corroborated some of this. It is well known that extreme female athletes have disruptions to their cycles. They may not cycle for months because there's a lot of other things too. They think with body fat composition, but that strenuous exercise is definitely impacting it. So we know there's something there, that exercise is impacting the female reproductive cycle. And then we see it here in horses. Now, to get to the big data, the horse embryo transfer data over the 2 years, in essence, we cut their fertility in half. So my recovery rate of non-exercise mares was about 63%. That means 6 out of 10 embryo flushes I would attempt, 6 times I would get an embryo. And that's average. Average is anywhere from 50 to 75%. Stephen Vogelsang, Katrin Hinrichs, who've done this for years, 3 out of 4, they're getting an embryo. Chris, it's 6 out of 10, or 3 out of 5 is getting an embryo. And again, that was my first time doing it. Or I did a few practice sessions, but then once we'd started the study. So we did that. When we looked at the exercise, so we went from 63%, that dropped to 34%. So again, 3 out of roughly a little bit over 3 out of 10 times on an exercise mare, I would get an embryo. So again, cut their fertility in half. What else was really striking and really exciting and nerdy, and I'm sorry if this gets a little nerdy, but it is really like the miracle of life. That's why reproductive physiology is so incredible. The embryo grades. Okay, so an embryo quality grade 1, perfect embryo, no imperfections, beautiful. They're just so beautiful under a microscope. Wow, that's the one we want to get. We want to transfer to a mare. Embryo quality 2, okay, you have a few little, they call them extruded cells. These are cells that died, and the embryo is really good at getting rid of those. So on the periphery, you see this little black mass. Embryo quality 2, they're okay. You can still transfer them. Embryo quality 3, wow, something's not going right. Half the embryo's dead, so half the cells are black and dark, half the cells are viable. You're like, oh, okay. And then grade 4 is pretty much a dead embryo, black throughout. Now, in our non-exercise mares, about 75% were embryo quality grade 1, and then 5% 2, 10% 3, and then 15% 4. I don't know if that adds up to 100. I rounded up a little bit. With the exercise, again, this is where the data was striking. Again, I got fewer embryos, so cut in half, and then a third were quality grade 1, and then just under a third were quality grade 2, and then a third were quality grade 4, just black, dead, nothing there. So that made waves. Popular media picked it up. I had some press articles written about it. When I went and presented it to a conference, and Professor Twink Allen stood up and said, "Young man, that was a beautiful presentation." And I was like, wow, he's one of the grandfathers of horse reproduction. I realized, wow, my PhD dissertation, you made waves. And then I tell this story. At a conference a few years later, I had a veterinarian from Germany very upset at my research and challenged me on everything. And I was really taken back. It was the first time anybody challenged me on this research. Everybody was so interested in it. And I just wasn't prepared. Again, was still young professor. And in reflection, I realized I was probably hurting his business because this had made news around the world in the horse, especially in the horse world. And he did a lot of embryo transfer and competition mares. And the owners were like, why am I paying you this money? We're not going to get an embryo. So he was starting to lose some business. So he was a little upset. Now, one study doesn't make a truth. I preach that all the time. And I did have some other independent labs around the world try to repeat some of this. They saw the same phenomenon. And then I went off to Clemson for my first professor job and repeated this work. So I did a follow-up study with Dale Kelly. He was my first graduate student. He is now Dr. Dale Kelly. He did his PhD with me at University of Florida. Then he went and got his DVM, and now he's a professor at Oklahoma State University. Brilliant person, just incredible, so smart and driven. And I was just blessed to have him as a grad student. But in his master's work, I said, okay, Dale, we need to look at the hormones because it's something I didn't look in my dissertation work. I suppose exercise is having an effect on reproductive hormones. So we went and repeated the experiment, but we were exercising them in the cool morning, not in the hot afternoon. South Carolina, around Clemson, that's the upstate. It gets warm in the summer, but not baking like Florida or Texas. It was warm, but we just kind of wanted to remove that element of heat and just keep it exercise and do what horse owners are probably typically doing, and that's training in the mornings. And again, when we went back and looked at the data, exercise mares had longer cycles. They had delays to ovulation. Then when we looked at hormones, the one hormone that stuck out the most was luteinizing hormone. That spikes with estrogen as the mare gets ready to ovulate. That's the one responsible for ovulation, and then for some other things after. But that's the one that helps really mature that oocyte. That was lower in our exercise mares. So we published that work. Exercise affects both ovarian follicular dynamics and hormone concentration in mares in 2011. So I think that was one of Dale's first papers. And it just was a very, very exciting study. So then I did another study after that. Okay. And this was with Rachel Smith. She was doing her master's with me. Great student, very hard worker. And we wanted to repeat what I did in Texas, but she was doing the embryo collections. We were exercising them in the middle of the day. So I wanted somebody, not me, doing the embryo collections or an independent, somebody else. So she was doing all of that. The middle of the day temperatures, again, 25 to 30 degrees C, 77 to 89 degrees Fahrenheit, humidity about 50%. So again, not very hot or very strenuous, but I introduced Doppler ultrasound. Now, for some women that have gone on to, or some of you listening, that maybe you've gone to ultrasound scans in humans during pregnancy, and they show you while they're ultrasounding, they'll push some buttons and you see some blue and red of the blood pumping. That's pretty common today across the world, Doppler ultrasound. Back in, this is 2007 and 8, Doppler ultrasound as far as livestock use or in horses was real new technology. So the handheld Doppler ultrasound I was able to purchase. And what we were looking at was the blood flow around the dominant follicle. So again, without explaining too much with reproduction, as the mare's ovaries generate follicles, they generate multiple, there becomes one that becomes dominant. And that's the one that's going to ovulate her oocyte. Well, there's a lot of angiogenesis. So if I use this, a lot of vessels growing in that follicle to pump the nutrients, pump hormones to that fluid that has the oocyte or egg in it. So it's called angiogenesis. So it's a generation of blood vessels growing there. And you can see this on a Doppler ultrasound. With a dominant follicle, you can see all the blood pumping around it. And so I was curious, okay, well, we know from Dale's study and my study that maybe we're disrupting that blood flow to the follicle, and that is having some impact on why I didn't get twice or the same amount of embryos, because that's where you might see an impact. Is it the oocyte or the early embryo? Where is that breakdown happening in these exercised mares? Is it poor oocyte quality, or is the embryo dying really young and we just never see it? So sorry, that gets a little into too much of the repro side of it, but to kind of explain this. So get to the results. Again, very exciting. So Rachel, her recovery rate was 67%, so a little bit better than mine, but I trained her, so I can take some credit. No, she did a wonderful job. 67% of her embryos recovered from control mares. They're 93% quality grade 1, and then only one embryo, it was 7%, had a quality grade 2. So to me, that says either different mares, different genetics, or maybe that heat in Texas had some impact because it was hotter there. Then we went to full exercise, 67% down to 43%. So not quite as drastic as mine, but significant reduction. So you did see that reduction. And then she, quality grade wasn't as bad either. It was 78% in quality grade 1, 12% in quality grade 2, and then she had some unfertilized oocytes that she collected. So overall, looking at that, there was an impact. It just was not as drastic as my PhD work, which we could explain that these mares weren't exercising in hotter or more humid conditions. So, but again, and then we took pictures of all the embryos. It's a really good study. Impacts of moderate exercise on ovarian blood flow and early embryonic outcome in mares. That was in the Journal of Animal Science in 2012. And then when we looked at blood flow, that was really exciting. Our control mares, their dominant follicles had really good blood flow around it. And then our exercise mares had really poor blood flow around their follicles. And then we did some correlation statistics that, yeah, if there was greater blood flow around the follicle, that usually meant we would get an embryo. So it was good quality. And again, that's contributing to the field of reproductive medicine and technologies in horses. So the take-home message of all of that, in horses, okay, again, that's where the popular press picked it up. Oh, could it impact women? You can't design an ethical study in humans to measure exercise and then flush embryos from a woman to see if she would get pregnant. There are studies out there looking at exercise in women during pregnancy, early pregnancy, mid-pregnancy, late pregnancy. Speak to your physicians. If you are exercising, wanting to get pregnant, or you are exercising and pregnant. Because I can't speak to that. There is a lot of research in that area. I can speak to horses, and I can say, because I'm probably the world expert in this field, exercise in early pregnancy can be detrimental to the mare, period. End of story. And it's not just my research and research we did at Clemson University. Other independent laboratories around the world have done science testing, universities. They're seeing the same phenomena. Many veterinarians that work in embryo transfer around the world have seen the same phenomena. So it does impact their cycles, and it does impact that early embryo's ability to survive. So generally what I would tell horse owners or people is, if you are trying to breed them during those days leading up to ovulation and post ovulation, very light exercise. Do not push her, and let her rest a little bit. And I think that would help improve your chances of getting an embryo. But again, I haven't done that study. and we'd have to follow it up with some research. Okay, what about the boys? Get to the end of it, the boys are fine. Those testes thermoregulate just fine. Dr. Vogelsang, Dr. Clay Cavender, Texas A&M did some follow-up studies. I remember Dr. Vogelsang told me he did a study where they put diapers on the stallions to try to raise their testes. It had no effect on their parameters when they went to collect them. Sperm cells were fine. Counts were fine. Those testes do a really good job of keeping them cooler than body temperature. So remember, cryptorchids, that's, let's say both testes are in the abdominal wall of the stallion, they're infertile because it's the testes are at body temperature and sperm cells can't be produced. That's why they're external. They're cooler than body temperature, and they do it just fine. So it has no impacts. The final study I want to talk about, and this is Dr. Jason Anton now. He was one of my master's students, and he went off to get his DVM. And it was exercising the pregnant mare from day 16 to day 80 of gestation, just to see if there's something there. And we published this in Journal of Equine Veterinary Science in 2014. We wanted to see if there was some impacts on reproduction. Again, we were using Doppler ultrasound, looking at fetal measurements, and, because again, the research in humans shows that, women can exercise during pregnancy as long as it's moderate to light, and there's usually no adverse outcomes. Horse owners can exercise their horses until the mare gets a little bit later in pregnancy, and then they'll probably let off or completely stop it. So that's why we kind of wanted to look at this and see if we noticed anything. And no, everything was fine. All mares went on to give healthy foals. The only thing that we found interesting was the mares had lower progesterone levels, which is important for maintaining pregnancy. But again, all these mares went on to deliver healthy foals. But we did notice that. And then the foals were actually growing larger in her. We were able to do some transabdominal ultrasounds and look. And I think we also did some rectal ultrasounds. But we measured the fetal length and fetal size. And these mares that were exercised, their fetuses were growing a little bit bigger. So maybe there was some good blood flow going on. Maybe exercise staying fit was healthy for the foal. I mean, that's what we kind of supposed. It wasn't detrimental at all. We didn't see anything detrimental, but maybe it was a little bit healthier and stimulating some growth in that. So again, need more follow-up research. But it was very interesting work. And, just to kind of tie this all up, it's something that I think you need to discuss with your veterinarian if you are looking to do embryo transfer in horses or whoever you're working with in the breeding barn to breed these mares. My advice, again, is if they are in a training program or competition schedule, try to schedule when she's going to ovulate and those early days of the embryo that they're either rest or very light for her. We can manipulate the mare's estrous cycle relatively easily. We start with light in the winter, and then we do have a lot of pharmaceutical drugs. We can short cycle them. We can prolong their cycles. So there is some way to manipulate it to a degree. So if you do have a big competition that you won't miss it, but maybe after she gets back, you kickstart her cycle and try to get an embryo out of her. I would argue we need to keep researching this because the mare's genetics are just as important as the stallions. They're critical to, the longevity of our horses, the health of our horses. We want foals that can withstand the stressors that are placed on them as they grow into competition, and we want these horses to live long and happy lives, and the mare's genetics are critical part of that. So we need to find ways to keep their genetics flowing throughout our different breeds of horses around the world. And embryo transfer is one of those ways that we can greatly increase that. So very interesting research. Loved it. Again, if you have any questions or any podcast topics that you'd like to see on video or listen to the audio podcast, always email me at podcast@madmartin.com. Check us out on social media, and please hit that like and subscribe button. I know you hear that on every YouTube video, but that helps us grow. And when we see more people subscribing to this, making nice comments, it just tells us, okay, we need to keep producing this content for you. So thank you so much for listening, and take care.